So NX-5948 is a unique molecule. It's something called a BTK degrader. And this is a new class of drugs that actually uses E3 ligase to lead to ubiquination and sort of destruction or proteasome degradation of the entire BTK enzyme. This molecule was developed really to meet that unmet sort of clinical purpose for those patients who progress after traditional BTK inhibitors or BCL2 inhibitors, a growing group of patients that are getting exposed to these drugs in earlier lines of care. So this was a phase 1A, 1B clinical trial that looked at patients with both CLL and NHL. The abstract that's being presented tomorrow is going to be focused specifically on CLL, and it's a dose escalation study followed with preliminary outcome data. What's interesting about this molecule is that it was given to patients with multiply relapsed CLL. The median lines of treatment was 4 to 5 prior lines, and these patients were actually very sick. Some of them actually had CNS disease. There were 5 such patients, which is a rare finding in patients with CLL, and more so they were heavily exposed. So nearly every single patient had been exposed to a covalent BTK inhibitor. About 80% had also been exposed to BCL2 inhibitor, and some had even seen non-covalent inhibitors. Also, a lot of them had baseline BTK mutations, which led to resistance to some of these other drugs. When NX-5948 was given to these patients, we saw robust clinical responses. And in a preclinical model, we can actually see that regardless of the mutations present, you see degradation of BTK occurring sort of in a rapid timeline. This led to an overall response rate at the primary endpoint, which was 8 weeks after starting, of 75%. But for the subset that we were able to follow even longer, we saw evolving responses with the overall response rate increasing into the lower 80s. And so this is a very promising drug. Durability needs longer follow-up. And so we're going to continue to follow these patients to see how they do with extended time on this therapy. But given the patient population of having refractory CLL that had progressed after prior treatments, this is a very promising treatment strategy to use a BTK degrader rather than the inhibitors that we're used to using in this relapsed refractory setting. Lastly, just a comment about safety, because that's always important when doing a phase 1 clinical trial. There were very low rates of grade 3 or higher toxicities. Only 1 CLL patient had to stop drug because of a treatment emergent adverse event. And overall, a really favorable safety profile with low risk of traditional BTK toxicities. In fact, no patient developed new atrial fibrillation while on this drug. Although, as I said earlier, we need longer follow-up to see if over time new toxicities develop. But in summary, I think this is promising data. We're excited to share this and hope to move this class forward and become another treatment option for those patients who have relapsed refractory CLL.